Hello everyone and welcome to the next session of the lecture series, and today's topic is respiration in plants. And today we'll be solving all the NEET previous year questions. Okay, and this is the continuation of the previous lecture, which was again based on the NEET previous year questions. Okay, so let's start with the first question. So here they are saying that the chemiosmotic theory. Okay, what does chemiosmotic theory tells you about the generation of ATP, how ATP are generated by breakdown of the proton gradient. Okay, so chemiosmotic theory of ATP synthesis in the chloroplast and mitochondria, okay, is based on, all right, options are membrane potential, accumulation of sodium ions, accumulation of potassium ions, and option D is proton gradient. So what happens during formation of ATP? There is a downhill transfer of electrons. Let's talk about mitochondria. If this is the inner membrane, okay, here we have the matrix. Here we have the intermembrane space. Okay, all the complexes are present here. Complex 2 and complex 4. And then finally we have complex 5, which is known as ATP synthase, which is involved in the ATP generation. Okay, so initially what happens, as the electrons flow from one complex to another, protons are here in the matrix. Okay. Okay, as the electrons flow downhill, okay, from one electron carrier to another, okay, a proton gradient is created across the membrane. So protons from the matrix are transported to the intermembrane space. Okay, and then what happens later? The breakdown of proton gradient takes place. Okay, and what happens? This leads to the generation of ATP from ADP and inorganic phosphate. So what should be the correct option? Correct option is option D, proton gradient. The next question is, in Krebs cycle, FAD participate as electron accepted during the conversion of, okay, so let's talk about Krebs cycle. So what happens when succinate is converted into fumarate? Then what happens? FAD acts as an electron acceptor and it gets converted into FADH2. Okay, so what should be the answer? Answer is option C, from succinic acid to fumaric acid. The next question is, respiratory substrate yielding maximum number of ATP molecule is, okay. So let's talk about respiratory substrates. What are respiratory substrate? Any organic molecule which can be oxidized is known as respiratory substrate. Okay, usually glucose is a respiratory substrate. Okay, usually glucose is a respiratory substrate, but depending on the requirement of the organism, proteins can also be used as respiratory substrate. Fats can also be used. Okay, as well as organic acids can also be used as respiratory substrate. So what happens? So option B should be the correct answer. Maximum number of ATP is generated, okay, through glucose. What happens if ketogenic amino acid and amylose and glycogen are used? So these molecules are, okay, all these molecules will be converted into glucose first. Okay, and what will happen? ATP will be used for the conversion of these molecules into glucose. Okay, so maximum amount of ATP will be produced by glucose. Alright. The next question is, how many ATP molecules, okay, we have to calculate how many ATP molecules could maximally be generated from one molecule of glucose. Okay, if the complete oxidation means aerobic respiration of 1 mole of glucose, okay, to carbon dioxide and water. So they have given this value 686 kilocalories. Okay, and the useful chemical energy available in the high energy phosphate bond of 1 mole of ATP also they have given 12 kilocalories. Okay, so now let's write down what is given to us. So 1 molecule of glucose they are saying, or 1 mole of glucose. 1 mole of glucose, okay, yields 686 kilocalories of energy. Now it is also given that 1 mole of ATP yields how much energy? 12 kilocalories. Okay, so number of ATP, okay, molecules produced will be 686 divided by 12, which will be equal to 57. Okay, so answer will be 57. The next question is maximum amount of energy, okay, or ATP is liberated on oxidation of, options are fats, proteins, starch and vitamins. Okay, so now what happens when, let's talk about the burning of 1 gram of any organic molecule. So if we talk about proteins, okay, protein yields around 4 kilocalories per gram of energy. Okay, whereas carbohydrate, okay, carbohydrates, they yield energy around 4 kilocalories per gram. Okay, and then, okay, carbohydrate, starch is a carbohydrate. If we talk about fats, okay, fats are usually slowly digested. Okay, they are not easy to digest and they require lot of oxygen also. Okay, to burn down, lot of oxygen is required. More oxygen is also required to burn fats. Alright, but fat interestingly yields around 9 kilocalories per gram of energy. Okay, so what should be the correct answer? Vitamins, they don't give us that much energy. Okay, they are just absorbed by the body. So answer should be option A, fats. Okay, fats, okay, maximum amount of energy is released, okay, by the burning of fats, although it is very slowly digesting. Now next question is respiratory quotient for fatty acid they are asking. Now what is respiratory quotient? Volume of, or you can say it is the ratio of volume of carbon dioxide evolved during respiration. Okay, respiration we know carbon dioxide evolved, oxygen is consumed, okay, to the volume of oxygen consumed. Alright, so options are more than 1, option B is less than 1, option C is 1 and option D is 0. Okay, so what should be the correct answer? Answer is option B, less than 1. Again, what happens if we talk about fatty acids? Again, a huge amount of or more oxygen is required. Okay, more oxygen is required. So what happens? Volume of oxygen increases, okay, as compared to carbon dioxide. So RQ value will be always less than 1. This is less than 1. Okay, for organic acid, it is more than 1. Okay, RQ value for carbohydrates, okay, that are undergoing aerobic respiration. For carbohydrates that are undergoing aerobic respiration, RQ value will be 1. RQ value will be 1. For example, glucose, which is C6H12O6, okay, plus 6 O2 give rise to 6 CO2, okay, plus some water is released, 6 water is also released, plus energy will be released. Here RQ value will be 6 divided by 6, which comes to 1. Okay, and 0 will be for succulent plant. Okay, because what happens here, oxygen is not evolved, only carbon dioxide is not evolved, whereas oxygen is consumed. What should be the correct answer? Answer is option B. The next question is, in which one of the following processes carbon dioxide is not released? Okay, so options are aerobic respiration plants. Okay, but what happens during aerobic respiration means they are talking about first glycolysis, then they are talking about oxidative decarboxylation and Krebs cycle. But we know that carbon dioxide is released during aerobic respiration, both in animal and plants. Okay, here also carbon dioxide is released. Alcoholic fermentation. So what happens during alcoholic fermentation? Pyruvate, which is produced at the end of glycolysis, is a 3-carbon compound. So this is first converted into acetaldehyde. Okay, now acetaldehyde is a 2-carbon compound. So carbon dioxide is released here, and acetaldehyde is finally converted into ethanol. Okay, again a 2-carbon compound. So here carbon dioxide is released. Now what happens during lactate or lactic acid fermentation? Pyruvate is converted into lactic acid or lactate. Alright, so these are 3-carbon compounds and no carbon dioxide is released. Okay, so what should be the answer? Answer should be option D. Lactate fermentation, no carbon dioxide is released. Now coming to the next question, the energy releasing metabolic process. Okay, energy releasing metabolic process in which substrate is oxidized, okay, without an external electron acceptor, okay, is called. So they are talking about the external electron acceptor. So what happens during aerobic respiration? What is the final stage of aerobic respiration? Terminal oxidation. It is the final step or final stage of aerobic respiration. And what happens during terminal? Terminal oxidation. Oxygen, okay, accepts electrons and protons and it's converted into water. So oxygen here is a external electron acceptor. Okay, but here they are saying that without an external electron acceptor. The first is photorespiration, which is not true. Aerobic respiration, there is an external, okay, electron acceptor, which is known as oxygen. Alright, then glycolysis is also not true. Okay, during glycolysis, NAD is converted into NADH. So NAD is the electron acceptor. But what happens during fermentation? Okay. Now let's talk about alcoholic fermentation. Okay. Pyruvate is converted into acetaldehyde first. Okay. Now this acetaldehyde is known as the internal electron acceptor. Okay. This is internal electron acceptor. Okay. And finally acetaldehyde is converted into ethanol. And carbon dioxide is released as a byproduct. Okay. So what should be the answer? Answer is option D. Fermentation takes place without an external electron acceptor. Now coming to the next question, the chemiosmotic coupling hypothesis of oxidative phosphorylation proposes that ATP, adenosine triphosphate, is formed because these are the options. ADP is pumped out of the matrix into the intermembrane space. This is not true because protons are pumped out into the intermembrane space. Next option is high energy bonds are formed in mitochondrial proteins. This is also not true. Mitochondrial proteins they are saying. Next option is there is a change in the permeability of the inner mitochondrial membrane towards adenosine diphosphate. This is also not true. This is no change in the permeability. The last option is the proton gradient forms across the inner mitochondrial membrane. So this option is correct. A proton gradient is formed across the inner mitochondrial membrane when there is a downhill transfer of the transport of the electrons. Okay. So correct option is a proton gradient forms across the inner membrane, and the breakdown of this proton gradient, okay, what it will do will lead to the synthesis of ATP. All right. And how does breakdown of proton gradient takes place? By the movement of the protons from the intermembrane space to the matrix. Okay. Protons from the intermembrane space goes back to the matrix through complex 1, which consists of F0 and F1 part. Okay. Here is the intermembrane space. Here we have the matrix. This is the inner mitochondrial membrane. Okay, protons flow through this F0, which is known as transmembrane ion channel, and then there is a conformation change in the F1, which leads to the formation of ATP from ADP and inorganic phosphate. Alright. The next question is, in alcoholic fermentation, what happens? Triose phosphate is the electron donor, while pyruvic acid is the electron acceptor. This is not true. Pyruvic acid is not the electron acceptor. It is acetaldehyde. Next option is triose phosphate is the electron donor, while acetaldehyde is the electron acceptor. So this is true. Now next option is oxygen is the electron acceptor. This is not true. Oxygen is not involved during alcoholic fermentation because it is an anaerobic process. Okay, it is an anaerobic, doesn't require oxygen. Oxygen is the acceptor in the aerobic respiration. Next option is there is no electron donor. This is also not true. Okay, triose phosphate is the electron donor. So what should be the answer? Option B is the correct answer. The next question is, how many ATP molecules are produced by aerobic oxidation of one molecule of glucose? Okay, now we know that one molecule of glucose Okay. Complete oxidation requires which step? Glycolysis, which produce 2 ATP and 2 NADH. Then we have the oxidative decarboxylation, which produce 2 NADH. Then we have the Krebs cycle. Krebs cycle produced 2 GDP, okay, which is equivalent to 2 ATP. Then we have 2 FADH2 are produced. And what happens? Along with this, 6 NADH is also produced. Okay, now we know that 1 NADH is equal to 3 ATP. 1 NADH is equivalent to 3 ATP. And 1 FADH2 is equivalent to 2 ATP. So what happens in glycolysis? Here we can multiply this with 3. Okay, here again we can multiply this with 3. 2 FADH2, multiply it with 2 because 1 FADH2 correspond to 2 ATP. Again here we can multiply it by 3. So here ATP formed during glycolysis. During glycolysis, how many ATP will be produced? 2 plus 6 equal to 8 ATP. Then if we talk about oxidative decarboxylation, what happens here? 6 ATP will be produced. Okay. Then we have the Krebs cycle. Krebs cycle, 2 ATP plus 4 ATP from FADH2 plus 18 from NADH. Okay, and what is the total? Total is 24. 18 plus 2, 20, and then 24. Now, if we add all the ATPs, 8 plus 6 plus 24, what should be the answer? 8 plus 4 is 14 plus 24 is equal to 38. What is the answer? 38 ATP are produced by one molecule of glucose. The next question is, what is the net gain of ATP? Here they are asking about the net gain. Okay, when each molecule of glucose is converted into 2 molecules of pyruvic acid. So here they are talking about glycolysis. Okay, so what happens during glycolysis? In the first step, preparatory phase, ATP is consumed. Okay, when glucose is converted into glucose 6-phosphate, 1 ATP is consumed here. So we will write cross, 1 ATP is consumed here. When fructose 6-phosphate is converted into fructose 1,6-diphosphate, then again 1 ATP is consumed. Okay, then later what happens? When 1,3-diphosphoglycerate, okay, is converted into 3-phosphoglycerate. Then here what happens? 2 ATP are produced. Alright, 2 ATP are produced here because there are 2 molecules of 1,3-diphosphoglycerate. Now what happens later? When phosphoenol pyruvate is converted finally into pyruvate. Here also 2 ATP are generated. Okay. So 4 ATPs are generated. Okay. 2 ATP were consumed, which give rise to 2 ATP. Okay. So net gain should be 2 ATP. Option B is correct. The next question is respiratory quotient value for tripalmitin. So tripalmitin, they're talking about the lipids here. So what happens? Tripalmitin. If we talk about the burning of tripalmitin, or when lipids are used as the respiratory substrate, and this should be the equation. It uses a huge amount of oxygen to burn. Okay, carbon dioxide produced is less. Okay, water is also produced, plus energy will also be produced. So RQ value will be carbon dioxide released divided by oxygen consumed, which comes to around 0.7. So what should be the answer? Option B, 0.7. The next question is fermentation in anaerobic. Okay, fermentation is anaerobic production of what? Protein and acetic acid. Not true. Okay, alcohol, lactic acid or similar compounds. So this is true because anaerobic respiration, okay, is of two types: alcoholic fermentation and lactic acid fermentation. And the products forms are ethanol, which is an alcohol, and lactic acid. Third option is ethers and acetone. This is also not correct. Fourth option is alcohol and lipoproteins. This is also not correct. The correct option is option B. The next question is life without air means life without oxygen would be reductional. Okay, not true. Free from oxidative damage. Again, not true. Impossible. This is also not true because life can survive without air. We know, like in prokaryotes, anaerobic respiration can take place. Okay, and humans in muscle cell. In muscle cells also, lactic acid fermentation takes place during oxygen depleted condition. Okay, lactic acid fermentation also takes place in lactic acid bacteria. Okay, so life will be anaerobic. Okay, so option D is the correct answer. The next question is fermentation product of yeast. So in yeast is a unicellular eukaryote. And in yeast what happens? Alcoholic fermentation takes place. And what happens during alcoholic fermentation? Pyruvate is converted into pyruvate, which is the end product of glycolysis, is finally converted into ethanol. Okay, which is a 2-carbon compound, and carbon dioxide is released as a byproduct. So what should be the answer? Answer is ethyl alcohol and carbon dioxide. The next question is, when one glucose molecule is completely oxidized. Okay, so they are talking about the aerobic respiration. Means they are talking about glycolysis, oxidative decarboxylation and Krebs cycle. So what happens? We have already seen that 38 ADP molecules, okay, will form 38 ATP molecules. Okay. Now they are also saying that 36 ADP molecules will get converted into 36 ATP. Okay, which will be correct when we will also consider that 2 ATP were lost. Okay, 2 ATP were lost during the transfer of NADH. Okay, from the intermembranes, from the cytosol to the matrix. Okay, so here option B will be correct. Option C is also not correct. Option D is also not correct. Okay, so option B is correct. 38 ATP will be produced from 38 ADP. Alright. Now, next question is oxidative phosphorylation involves simultaneous oxidation and phosphorylation to finally form. So what happens again during oxidative phosphorylation? ATP is generated in the end. Alright, or oxidative phosphorylation, also known as, you can say, electron transport chain, also sometimes called as terminal oxidation. Okay, where oxygen is the electron acceptor. Okay, final electron acceptor. And what happens? ATP is generated during this process. Alright. So oxidative phosphorylation, how you can define oxidative phosphorylation? You can say that the energy, okay, of the oxidation reduction reaction, is used for the formation of ATP. Okay, so correct answer is option D, ATP. Next question is, here they have given you this figure. Alright, so now what they are saying that 3 boxes in this diagram represent 3 major biosynthetic pathways in anaerobic respiration, and arrows represent the net. Okay, arrows represent the net reactants and the products. Okay, so these are the pathway they are saying, pathway A, pathway B, and pathway C. Now what we have to do is that they have also given that it is starting with glucose. Okay, so if it is starting with glucose, then they are talking about glycolysis first. Alright, and what we have to do, you have to figure out what is 4, 8, and 12 here. Okay, now pathway A, now we can say that it is glycolysis because it is starting with glucose. Now pathway B, we can say after glycolysis what we have? We have the Krebs cycle. Okay, and pathway C, you can say, can be, okay, or should be oxidative phosphorylation. Okay, or you can also say electron transport chain, or you can say terminal oxidation. Alright, now what happens? You are seeing here these arrows. Okay, these arrows are indicating that what is the output. Okay, and these arrows you can see are the input, which is, okay, what molecules are going, okay, are used in these pathways, and these arrows you can see are the output. All right, so what is the common thing? What is the common output of these pathway, glycolysis, Krebs cycle, and oxidative phosphorylation? ATP are produced during these pathways. Okay, and these are all the inputs, okay, which are going into these pathway. So option 1, this one can be any of the intermediates of glycolysis pathway which is entering here, and it will lead to the production of pyruvate here. Second can be pyruvate. Okay, this fifth can be acetyl coenzyme A. Okay, this can be acetyl coenzyme A from any other source other than pyruvate, other than, you can say, pyruvate, or other than glucose, okay, such as fatty acids. Now option 2 can be pyruvate, because after glycolysis pyruvate is formed. Alright, now pyruvate will get converted into acetyl coenzyme A, then it can enter the Krebs cycle. Okay, then what happens during Krebs cycle and during glycolysis? What are produced? NADH is produced. Okay, and FADH2 is also produced during glycolysis. So 6, 7, these can be NADH and FADH2 which are produced during the glycolysis and Krebs cycle. okay, entering into the oxidative phosphorylation pathway. And again 9 and 10, again these can be NADH or FADH2, okay, from any other source. Okay, and what happens? 11 can be water, or should be water, you can say, because this is terminal oxidation. Oxygen is the acceptor of the electrons and protons, and water is produced in the end. But what we have to do, we have to figure out what is 4, 8 and 12. Okay, so options are, is it water, FAD, FADH2, NADH or ATP? Alright, so what should be the answer? Answer should be ATP. Okay, option 4, 8 and 12 should be ATP. Okay, so this was the end of this lecture. Thank you very much for listening.